Hello and welcome back. We're going to talk in a little more detail in this video about the different types of microscopes. So I said in the previous video, one of the types of microscopes that are out there are light microscopes. And it's called a light microscope because you are using essentially a light, whether it comes from underneath the sample or from on top of the sample. You're using light to be able to view whatever your sample is. Okay? And light microscopy allows us to see the shape of different cells. It also allows you to see some structure, although not in great detail. All right? When we're talking about a light microscope, the main organelle that you're going to be able to see in a eukaryotic animal cell would be the nucleus. All right? If you're looking at a plant cell, you can also see things like chloroplasts, which are the green components of the cell. That's where photosynthesis occurs within that plant cell. And you will also be able to see a specialized structure known as a vacuole. All right? And that's really just kind of a large storage chamber for the plant cell. So we can see some detail inside the cells using a light microscope, but really it gives us an idea of overall shape of the cell. With bacteria, again, you're going to be able to see the bacteria, but you're not going to see a lot of detail associated with the bacterial cell. Okay? Light microscopes, typically the max magnification is usually 1000x. So if you're looking at a sample 1,000 times bigger than it is in real life. All right? And typically the resolution is going to be 0.2 micrometers. All right? So if something is 0.1 micrometers apart using the light microscope, you're not going to be able to see those two structures as being different. You're going to see them kind of as one image. So our limit on resolution is about 0.2. Our max magnification is about 1,000. Okay? Electron microscopy is going to use something different for you to be able to visualize the cells. All right? Electron microscopy requires the samples to be coated in metal, which is why electron microscopy is not compatible with being able to view live cells, because once you coat that cell with metal, it's no longer living. Okay? So you've got to coat the cell with metal, and then you're using electrons. And what that electron microscope does is it shoots and bombards the sample with electrons, and the electrons are going to kind of bounce off of the metal, and how they bounce off the metal is interpreted by the computer associated with the electron microscope and is translated into the image that you see associated with an electron microscope. Okay? And in terms of our max magnification, it's a lot greater. So we're talking about being able to visualize things 100,000 times larger than they are in real live life. Okay? And our resolution is also greater. Okay? The resolution for most electron microscopes, 2 nanometers. Okay? So things that are 2 nanometers apart, you're going to be able to distinguish as 2 distinct points. All right? So what you can see with an electron microscope, the electron microscope now allows us to visualize things that are very, very tiny, like viruses. We can see viruses. You're going to be able to see more detailed structure within a eukaryotic cell. So you're going to be able to visualize more of the organelles. So things like mitochondria. You're going to be able to see associated with the electron microscope. Endoplasmic reticulum would be another structure. Golgi apparatus, and I know these terms right now don't make a lot of sense to you because we haven't talked about the different organelles yet. But I just want to have that in there to kind of give you an idea. The electron microscope, you're going to be able to see individual components within the cells. Those individual components within the cells are the organelles. Okay? And the difference really between, because with our electron microscope, right, I said we had the scanning electron microscope. The scanning electron microscope allows you to visualize essentially the outside or the exterior of a particular surface or a particular sample. The one I have in the PowerPoints that I like, I think it's really kind of cool. They did a scanning electron micrograph of an ant. So you can see kind of the outside of the head of the ant. You can see some ridges and grooves that are associated with the exoskeleton of the ant. And those are things you wouldn't be able to see just looking at the ant with the naked eye. So the scanning electron microscope allows you to visualize the outside or the exterior of whatever your samples are. The transmission electron microscope, this requires an additional step, okay? You have to cut your sample into individual slices. So if you're thinking kind of like a loaf of bread, the scanning electron micrograph might be able to look at the outside of the loaf of bread. If you cut the loaf of bread into slices, the transmission electron microscope is going to look at the slices. So you get to see interior cellular structures. So when I talked about being able to see things like the organelles, being able to visualize the organelles, you're going to be using a transmission electron microscope. Okay? So that's kind of the overall basic differences between light and electron. All right? Remember, light, you can view live samples. Electron microscope, all of the samples that you're looking at are going to be no longer living. They'll be dead. All right? And there's a difference in the magnification and the resolution associated with the two. All right? But they're really great tools to be able to visualize what's happening inside of the cell. If you have any questions, please let me know.